Okay, we've spent the past few lectures talking about the digestion of the major biomolecules, carbohydrates, fats, and proteins. So let's now talk about the fate of these biomolecules. That is to say, what happens to them once they reach the bloodstream. And the same really is the case with all of them. If they're needed for energy, they're going to be utilized for energy. Certainly, carbohydrates are going to be our main source of energy. If they are needed for synthesis of biomolecules within the body, that is going to happen as well. And if both those needs are met, energy and synthesis, storage of the biomolecules is going to occur. Glucose is the only fuel for the brain, so that is going to be the number one priority for glucose. Now, that's not totally true. During times of starvation, our brain does utilize ketone bodies, and we'll talk about that shortly. But the brain does not use fatty acids, and it does not use amino acids to make ATP. So glucose is really the only fuel the brain uses during normal times. And I put quotes around normal because everyone has their own normal. Glucose is going to be used for metabolically active cells to make ATP. If both those needs are met, glucose is going to be stored as glycogen or as fat. If it's stored as glycogen, it undergoes the process of glycogenesis, which is making glycogen. The main stored sites of glycogen in the body are the liver and skeletal muscle. Other cells have storage of glycogen, but they're very small quantities. So the main storage sites of glycogen are skeletal muscle and the liver. But there are only limited amounts of storage for glycogen in the liver and skeletal muscle. There's much more storage in adipose tissue, so that glucose can be converted to triglycerides to be stored as adipose tissue via the process of lipogenesis. Fatty acids can be used for energy as well, and we'll talk about that process. If they are not being used for energy, fatty acids are going to be stored in the form of triglycerides in adipose tissue. And then we have proteins, specifically amino acids, which amino acids are absorbed into the bloodstream and they're going to be utilized for protein synthesis. We have proteins throughout the whole body. They are ubiquitous within our body. Channels are made of proteins, pumps are made of proteins, hormones are made of proteins, enzymes are made of proteins, fibers, collagen, muscles, all of these are made of proteins. So there's quite a wide breadth of things within the body and molecules that can be made via the free-floating amino acids within the blood plasma. Keep in mind, everything we're talking about here is initially in the blood plasma because that is where it has been absorbed from the GI tract. Amino acids, in the absence of a sufficient amount of glucose in the body or sufficient amounts of glycogen, amino acids can be converted to glucose via the process of gluconeogenesis. And that will allow for the creation of more ATP. And we'll talk about that process as we move forward. So when we eat, we are in what's known as the fed state. When we don't eat and haven't eaten for hours, that's known as the fasted state. So during the fed state, we have increased blood plasma glucose concentration. And as we talked about the very first lecture of this semester, the increased blood glucose levels is going to stimulate the release of insulin from pancreatic beta cells and is going to allow for glucose uptake throughout the body. And this is also going to promote glycogenesis, the production of glycogen molecules, the glycogen polysaccharide. So during the fed state, insulin is going to promote glycogenesis. It is going to inhibit glycogenolysis. Glycogenolysis is a process of breaking down glycogen to increase blood glucose levels. Keep in mind, during the fed state, we theoretically have sufficient amounts of glucose in our blood, and there's no point in breaking down that glycogen. We have another hormone from the pancreas release, and that is glucagon. It is released from the pancreatic alpha cells. Keep in mind, insulin's released from the beta cells, glucagon's released from the alpha cells. We have increased levels of glucagon during times of low blood glucose where we haven't eaten for hours. And this promotes glycogenolysis, that is, the breaking down of glycogen to move glucose into the blood to increase our blood glucose levels. And it inhibits glycogenesis, that is to say, it inhibits the building up of glycogen molecules. We do not want to build up glycogen. We want to break it down to increase our blood glucose levels. So let's talk a little bit more about glycogenolysis. Glycogenolysis breaks glycogen down into two molecules, into glucose and to a much larger degree, glucose 6-phosphate, otherwise known as G6P. It's mostly glucose 6-phosphate. The goal of glycogenolysis is to get glucose in the blood plasma, to increase the blood glucose concentration in the body. Some of that is achieved directly by the glucose during glycogenolysis, but glucose 6-phosphate in the presence of oxygen anaerobically is going to be converted to pyruvate. Anaerobically, it's going to be converted to lactate. Glucose 6-phosphate is an intermediate within the glycolytic process. So this is no different than that. In the presence of oxygen, it's converted to pyruvate. In the absence of oxygen, it's converted to lactate. In the liver, pyruvate and lactate are converted to glucose via the process of gluconeogenesis. Gluconeogenesis is the production of glucose from non-carbohydrate substrates. So this glucose 6-phosphate is going to eventually be converted into glucose, but via pyruvate and lactate. Okay, so let's talk about what happens when we break amino acids down. Amino acids can be converted to glucose via the process of gluconeogenesis. The amino acids can also enter glycolysis or the Krebs cycle. And keep in mind, when I say they're entering glycolysis or the Krebs cycle, the end goal there is to make ATP. In the process of breaking down amino acids, a step known as deamination occurs, which creates ammonia and the ammonia ion. Both of those are toxic to the body, so they're immediately converted to urea, which is eliminated from the body via the urinary system. Okay, so now let's talk about lipolysis. Lipolysis is the breaking down of adipose tissue to use it for energy. Adipose tissue is formed by triglycerides, which we looked at in a previous video. Triglycerides are broken down into glycerol, which via the process of gluconeogenesis turns into glucose. The fatty acids, those side chains, if you will, that we looked at, is converted to acetyl coenzyme A, and acetyl coenzyme A can then enter the Krebs cycle to make ATP. If acetyl coenzyme A is getting created faster than the Krebs cycle can utilize it, the acetyl coenzyme A turns into ketone bodies, which the brain can then use as a fuel source. Keep in mind, lipolysis is going to occur during the fasted state, during times of low blood glucose levels. We break the fat down, and these two processes occur. That's it for our semester for Cabrillo College. Corona Fall 2020. I know it's been a challenging fall for everybody, a challenging, I guess, last 6 months. And it will continue to be, but we're strong, resilient, and hopefully I'll see some of you on YouTube next semester, maybe on campus in 2022.